Welcome you in my assignment presentation. My name is Jean Damour Manirere, student from Ines Ruhengeri, in Faculty of Health Science, Department of Biomedical Laboratory Science. The module is Applied Immunology, and the lecturer is Dr. Francois Xavier Ndayambaji. So I'm going to present about topic 5, which say the principle of vaccination. And this topic have 3 questions, 16, 17, and 18. The 16th question say what makes vaccine effective. 17, how messenger RNA vaccine works. And 18th, why are adjuvants used in vaccination? So let's get started and dive in. We are going to start about this 16th question, which says, what makes a vaccine efficiency? Actually, the vaccine is said efficiency when it has ability to induce a strong and lasting immune response. That immune response produces a memory cells. Remember, the target of vaccine or vaccination is to induce those memory cells. There is some factor that can contribute to the vaccine effectiveness. Among those factors, we can say immunogenicity. What is immunogenicity? Immunogenicity is the ability of the vaccine to induce a strong immunity. Those strong immunity will end up inducing the memory cells, and those memory cells will eliminate when we fight against the harmful pathogen when will be exposed. The second factor is safety. The efficient or effective vaccine should not cause another disease or side effect. When the vaccine can cause another disease, it is not effective. The third one, say, duration of protection. Those production of memory cells, those memory cells should be able to resist for a long time. Another one is targeting the right antigen. The effective vaccine should contain a single antigen which induce immune response. So here there is a chart that can explain summary showing the effectiveness of a vaccine. Here you see this is a vaccine which contains antigens and adjuvants. When the vaccine is introduced in the body, the antigen are recognized by antigen presenting cells, those macrophage and B cells. Automatically, those antigen presenting cells present the antigen to the T helper cells, CD4 or CD8, according to the kind of antigen, because the antigen can be endogenic or exogenic. When it is exogenic, it is presented by MHC class 2 here, and when it is endogenous, it is represented by MHC class 1. So the T helper cells will automatically stimulate the cytokines which allow either B cell to be proliferated into plasma cell, and the plasma cell differentiate by producing antibodies and memory B cells. When it is endogenous antigen, the CD8 will allow the differentiation into T helper cells, no, into T, into memory T cells and cytotoxic T cells to fight or to eliminate those cells which are abnormal. I think this what I can say to the effectiveness of vaccine. The following question say, how do messenger RNA vaccines works? Actually, messenger RNA vaccine are the synthetic messenger RNA which seems like which can produce the protein. That protein looks like the protein of pathogen. So when when the body cells are instructed to produce that protein, this protein will be recognized, then induce the immune system, immune response. So when the immune response will be recognized, those proteins will automatically produce memory cells, which will fight when that pathogen will be exposed to that body. So here there is a steps of preparing or synthetic of messenger RNA vaccine. First of all, the first step is messenger RNA synthesis or vaccine itself preparation. Uh, they prepare, there is a diagram there that is going to show us how it is prepared. Then the second step is the introduction of messenger RNA. It means it is vaccination itself. Then the following is protein production. It means that messenger RNA will be translated into the protein which looks like the protein of virus or another pathogens. And the last one is the immune response. Automatically, when that protein will be produced, it will automatically induce the immune response. And the last one is the memory cells formation. Now, we can see the diagram that can explain where this. So here it is the pathogen. The scientists take the DNA of this pathogen and they inject in the vector so that DNA can be amplified, and they study the composition of this protein, and they synthesize the messenger RNA which looks like this protein. And when this messenger RNA is synthesized, it means that the vaccine is all ready. Now they inject this is the vaccination. It means the injection or administration of this messenger RNA into the body. It will go in the cells. When it will go in the cells, it will be translated into protein, and automatically this protein will induce the immune system. This is a good example of messenger RNA vaccine. Now, let's go to the last question, which says, why adjuvants are used in vaccination or in vaccine? Actually, the adjuvants are the substance, non-organic substance, which are added in the vaccine preparation so that it can induce or accelerate the ability of the vaccine to induce the immune response. Yes. So the adjuvants they can function in the following steps. it can enhance immune response, it can reduce antigen quantity. It means when there is advent, we don't need a high quantity of antigen. That's why they say it can reduce the antigen quantity. Even if it is a raw quantity of antigen, it can induce the immune response. They can improve memory cells. As you know, the principle or the objective of vaccination is to induce these memory cells, because it is the memory cells that will fight against the harmful pathogen when it